Thank you so much, Rob, for the introduction and for the invitation to speak here. I would have it's an honor. There's so much PopGen happening in Vienna, and it's a pity that I was not there in person. It was a big trip to fit in already lots of trips this spring, but I hope to visit soon. So please invite me again. Um, so today I want to tell you what we're doing in our lab, how we are using evolutionary principles to understanding species responses to global change. So I want to begin by traveling using Google Earth to understand the magnitude of the problem that we're facing. This is Brazil, a region in the Amazon, where we can see that since the 80s, there's been a progressive deforestation to the point that the region has lost 50% or 50% of the habitat has been transformed into for human use. And these were pristine Amazonian forests. Let's travel north now, again with Google Earth, to the region of the Columbia Glacier in Alaska. Same time series of images, now in a region that is not touched directly by human, but we see that there's lots of change. And this is indirect impacts from human-caused climate change, which probably you're noticing has caused the retreat of the glacier, but also is changing the distributions of species. You can see how plants are moving to higher altitudes with the melting of these high altitude ice. So plants and animals are migrating, tracking climate change. These are the two main causes of what we know is now an acceleration of extinction risk: climate change and land use change. A few years ago, Mark Urban did a meta-analysis of studies that tried to predict what might be the consequences of these human impacts in the probability of extinction of species. And what he found was that across studies, there was about an 8% of all species predicted to potentially become extinct. These were very heterogeneous studies, right? So that's why you see such a large variance from niche modeling to demographic modeling to others. And the other thing that he observed was that as climate change progresses, that is, the assumption of the models were stronger and stronger in terms of our climate change trend, the extinction accelerated. So we are entering a mass global extinction, which means we're in an era where maybe a large proportion of species might be extinct. However, none of these projections, studies, incorporated the possibility that evolution might save some of the species through evolutionary rescue, adaptation, nor maladaptive evolutionary processes that could generate a vicious circle and lead to an increased extinction risk. And the thing is that now with the molecular revolution, with population genetics now applied to natural populations, we know that evolution is not just a slow process that leads into the speciation of new species, but it's actually acting in every population all the time. And so these are some examples where through standing pre-existing genetic variation in a population, we have observed a rapid morphological or genetic shift in these populations that probably led to adaptation. So this standing genetic variation is key, and this evolutionary process is key, but myself and others have argued that this process is not yet well implemented in global biodiversity framework, where genetic diversity, the basic element of evolutionary processes, is weak in terms of its presence in sustainable development targets. So in our lab, we are trying to go very narrow in understanding evolutionary principles and then think very broadly how they could be applied to understanding adaptation and extinction in general across species. And I think that there are some key questions to be asked that are evolutionary questions that I'm going to try to ambitiously address today, such as what is the genetic basis that is standing in a species and the architecture, the genomic architecture of adaptation to an extreme environment, for instance, one that could be faced with climate change? Is there enough time for adaptation to take place? That is, do we know if adaptation is really rapid enough in the order of few generations? And is there enough land that we're maintaining to maintain enough genetic variation and adaptive potential for the future? So let's get started. To begin, I want to introduce our supermodel organism, Arabidopsis thaliana. You probably have heard of it or seen pretty pictures. This is a picture in its native environment among other annual plants. And this is a picture of a zoom out of a native environment. This one is not California, although it's very similar how ecosystems look here, but it's Spain, one of the oldest populations that we found, what we call relict populations that probably were there millions of years. And this is near my hometown. But of course, you've probably seen Arabidopsis in this native environment, not the previous one, which is our laboratory. It is the model plant for genetics, and we have lots of genomic resources. Although normally we study a single ecotype in the lab, that's led to the first reference genome and all the molecular resources. In our lab, we're trying to combine both. We are trying to combine the genetic tools that it has, the 1,000 genomes, and normative collection with the facts that it is globally or very broadly distributed across very steep gradients of environment, and we can do very large experiments with it. So, but where do we begin when we're thinking about potential adaptation to climate change? For this to happen, there has to be standing variation related to climate adaptation. So how do we get at that? This is actually a pretty old concept through common gardens. And it's something that was asked by Klaus and Kekin-Heisey in the Carnegie Institution at Stanford, which is where I started my lab 5 years ago. Now I have moved it back to the University of California, Berkeley, and Howard Hughes. And what they did was collecting populations of a bunch of plant species, in this case is Potentilla, and plant them in Stanford. And so they could see that from different elevations across the gradient of California, they could plant them in Stanford, and the low altitude did better. But not only did that, but transplanted it to 3 sites. So they did all reciprocal transplants. And what they saw is that typically the population that was collected most closely in environment of origin had the highest vigor, to the point that some populations failed to survive. And this is what they asked climate types. So with this elegant experimental design, by homogenizing the environment at one location, they revealed genetic differences of populations within that location. And the second is that they reveal that the highest vigor is the local one, suggesting that there has to be some evolutionary adaptation that is encoded at the genetic level for this climate adaptation. After them, many people have repeated this kind of experiments with hundreds of plant species, and 70% of them show positive signals of local adaptation. So this seems to be like a general phenomenon across species. So we wanted to leverage the same thinking with Arabidopsis, except for that we could add a genomic dimension to these experiments. So this is an experiment that I conducted during my PhD in Madrid, Spain, which is in the southern edge of the typical geographic distribution of Arabidopsis, although we know there are some others a little bit southern. And we transplanted 500 populations from the 1,000 genomes that have a broad distribution, as you can see here, and plant them at high replication levels in this experiment. Not only one, again, but in 2 locations in Germany, Tübingen, where I was doing my PhD, so that we could see contrasting adaptations to these environments. On top of that, under these foil tunnels, which only excluded rainfall, temperature was roughly similar to outside because they were opened. We could also impose a low regime of precipitation and a high regime of precipitation, simulating Spanish and German rainfall in all factorial combinations in the two locations. With this in hand, what do we see when we plan this model system in the wild? In hundreds of natural variants was a beautiful diversity. With these stressful conditions, you reveal a series of phenotypes, and some of them as striking at these rosette colors. But we were not focused on the charm of Arabidopsis in generating beautiful colors, but we were focused on fitness traits that would tell us something about whether a population was adapted to that environment by the fact that it would survive, which we track like with these pins, or not, and if it produced fruits, and then how many fruits they produced. Okay, so what do we see with these kind of experiments? I'm just summarizing it very shortly in a cartoon because this has been shown multiple times. With this data, we can see generally that there's large fitness variation, meaning survival and reproduction in an environment like Spain, with 60% of all the populations of this species not being able to make it. So this suggests a very strong local adaptation pattern. And it was typically the populations that had been collected from faraway environments following this local adaptation theory that could not survive or had lower fitness. And the same we saw in Germany, except for it's vice versa. The populations that come from a similar environment in Germany, which is a faraway environment in Spain, right, had the higher fitness versus the others. All the fitness differences were smaller. And this, again, has been shown in Arabidopsis and other species multiple times. What was different of our experiments, it was that it was the first time that we had whole genome data for these common garden experiments. So I thought of applying genome-wide association approaches, which normally we use to map phenotypes in the genetic basis of phenotypes, instead to use fitness with it. So what you see here is Manhattan plots, where in the x-axis we have all the SNPs in the genome, over 1 million SNPs that had been, carefully selected and quality controlled and frequency pruned. And then we have different environmental combinations. Chiefly the one that I've been telling you about, the strongest fitness difference was in Spain and low rainfall, and therefore you see many peaks associated. So what do we see from this? We see a highly polygenic architecture, as you can see. Nevertheless, a relatively high heritability, so up to 40%, which is pretty substantial for fitness traits. This is the nightmare of a molecular biologist, because how are you going to follow up the molecular basis of this? But I think the heaven of a quantitative geneticist, because it means that then we can use quantitative genetic thinking to understanding fitness in the wild. And so what general patterns we've seen, apart from this, one question that always emerges is whether this is repeatable. And one thing that was interesting was to look at the loci associated in some environments and compare it with others, and seeing if there is a signal that the loci are the same and whether it goes in the same direction or in opposite directions. And generally what we see is a pattern of antagonistic pleiotropy that in statistical tests show is more likely than conditional neutrality. What this means is that it is more likely that the same loci, at least big chunks in the genome, are associated in multiple environments, but the effect sizes go in opposite directions. And this we might expect because we expect that there is lots of fitness trade-offs, right, in ecological settings when investing in different traits, and plants are integrated systems. The good news is that because heritability is relatively high, it means that there might be fuel for adaptation to happen. And actually, this is not something that is private to Arabidopsis, but it's likely common in many species. And there was a recent meta-analysis quantifying heritability of a number of animals, even some that have relatively small effective population sizes. But this also brought up the question of, well, we know that there's heritability, but what can we do with these GWAS? If we cannot molecularly follow up certain loci to understand adaptation and try to make predictions, is this predictable at all? So after this, we created a new set of common garden experiments. These are at Stanford campus, precisely where Klaus and Kick and Heisey did the first common garden experiment. So that was a very momentous time of my career. In this case, we have a setup with 14 zones with different, let's see if I have the zones, yeah, different sections and rows, each of which is independently controlled by automatic timers to generate precipitation patterns with some sprinklers from top. And this was a massive endeavor from our lab, including PhD student Laura Leventhal, postdoc Megan Ruffley, and many lab members. So I'm very thankful to them. And these treatments, because we from previous experiments had seen the precipitation drove most of selection, but we didn't know exactly what precipitation was exerting the selection pressure. We created frequency by abundance, all fully factorial combinations. So we modified the total abundance, but also at what times they received. And we created this gradient of frequency by abundance. So to the right, the conditions became more scarce, right? Like the abundance was, the rainfall was scarcer, but also more erratic, such as like once a month only they got precipitation. And sometimes we also had precipitation rainout shelters. So this reduces 50% of the precipitation natural to California. Okay, so with this in hand, how could we test whether we had learned anything from previous GWAS? To test this, we use survival polygenic scores, which are the same polygenic scores that are used for human genetics, where we would take all the top alleles from GWAS of fitness in Spain, and then conducted this score, summing over the alleles that we considered adaptive, if they had a positive effect in fitness or correlation with positive fitness, or negative correlation with fitness if they were low adaptive. And once you sum this all, you create a Gaussian distribution, right, of your 1,000 genomes of Arabidopsis. Then we selected the top and the bottom, and then we compare them. So here's the survival results from the first year of these field experiments, where in a lowering rainfall and more stochastic rainfall, we see that generally populations decay in survival. Duh, we expected that. But what is very, very cool is that the populations that we had selected to have high polygenic scores versus low polygenic scores differed by about 20% of survival, at least in the most extreme precipitation treatments. And if we do a raw Spearman correlation between the polygenic scores and the actual survival scores in this experiment, and remember, this experiment is an entirely new experiment, right? None of this data was seen in the training set. The training set came from the Spanish experiments. We see relatively high predictive scores, which is probably enviable by human geneticists when they're trying to predict things that are complex, like neurological diseases or cardiovascular diseases and so on. So it looks like there's some predictability, and we, moving forward, want to try to understand better these complex polygenic architectures using new ways of thinking using CRISPR funded by NIH. In the meantime, as we validate some of the loci that are underlying this polygenic score, we thought, how can we use this to understanding potential differential population responses to global change? So we can get into the nitty-gritty of the details later. We are improving this, but in 2019, we made this projection, calibrating GWAS in Spain and Germany and trying to project what must be the loci that are adaptive in different environments, doing an interpolation, and then asking, do those plants have the right loci, or do they have the, quote unquote, bad loci, just like we had done in the previous polygenics course. And what we have seen is that populations in the transition zone from Mediterranean climates to temperate climates, which might right now be adapted to temperate climates, seem to have a lower polygenic score, meaning they might become maladapted in the future. And this coincides with a precipitation change in Europe that is decaying. So temperate regions are becoming more Mediterranean regions. For tourism, good. For Arabidopsis, maybe not as good. Okay, and we are working on improving those predictions, and you're going to see in a second what data we're going to use for that. But the conclusion of this section is that we've seen that the genetic basis of adaptation exists in Arabidopsis. There's plenty of standing variation. The architecture is polygenic, highly G by E, but it seems to be heritable to some degree and to some degree predictable. But this first part is just a Gedanken experiment of, will there be potential for adaptation, right? If there's fuel for adaptation. But that doesn't mean that adaptation will be realized, right? That the populations will actually evolve. And this is because there needs to be demographic dynamics happening. There needs to be enough time for it to happen. So that's the second question. At some point, when I conducted those previous experiments, I realized that we didn't have likely enough locations to train data sets to make future predictions, and also not enough time because we were evaluating fitness over a single generation, and we need to study evolutionary dynamics over time. So we thought of these, amazing, beautiful experiments. What will be the ideal experiments? We'll be collecting populations from many regions, right? So we start with this high standing variation, planting them in many, many regions, like all of this, across the geographic distribution of Arabidopsis, and doing that over time. So we started thinking about the feasibility of this experiment, and it actually became feasible through creating a network of collaborators to conducting this. So I co-founded this network together with Nick Heppens and François Vasseur in University of Frankfurt and in Montpellier, along with many collaborators across the globe. We call this the Green Nets evolution experiment. And it was a citizen science-like setup so that many people could join, where we generated seed mix that had high standing variation by combining wild-type Arabidopsis seeds. We send these seeds to collaborators along with sensors and experimental materials. So this is an Arabidopsis trait where you can have hundreds of Arabidopsis. And people sowed only one time these thousands of seeds in 12 subpopulations in their botanical gardens or backyards where they work outdoors. Then you can see some pictures. Depending on the outdoors, the populations, suffer different environmental stresses. And we did this in 45 locations, 32 locations survived. And over time, we've been collecting tissue samples every year, documenting them in distributed approaches with Google spreadsheets, with whole genome sequence flowers, 2,500 approximately pooled population samples from 3 locations, 3 years that we did, and by we, I mean a research technician in our lab. We've also created computational tools based off of RUPS population and other people's ecotype reconstruction approaches, which we've packaged in a pipeline with SnakeMake and a C++ library to conduct frequency manipulations very fast. And with this analysis team, we're now synthesizing what we found in GridNet. So let me take you sneak peek into GreenNet. Again, this is just one of the sites of 32 that survived. We have an evolving sequence experimental design, right? We started with a bunch of seeds that were highly genetically diverse. They experimented some stressors, some outdoor environments that generated fitness differences, right? Like natural selection. And then we sequence the survivors in a few generations down the line. What we see is that there is evolution happening in GreenNet. We see allele frequency trajectories that are parallel across independent replicates, which suggests that there's likely natural selection involved. And the rationale here is that we expect lots of allele frequency changes. But if it was drift driving them, if there's in independent replicates, then we would see that those allele frequencies will go in all directions. But if those allele frequencies go in the same direction across 12 independent populations in the same environment, like this green increasing or red decreasing, it's highly suggestive of natural selection. And the correlation in some sites is actually very high. So it's highly parallel selection. Through haplotype reconstruction, because we have the founders, which were the same in the first generation for every site, and for which we have complete whole genome data, we can reconstruct the relative proportion of those 230 founders that make up this ecotype frequency. So here, each of the 230 founders are color-coded by one color, and we can also see their relative increments, right? Like at the beginning, they're equal proportions, and then over time, they change and they're not equal proportions. And precisely these sites, which is something not very common, we saw a single ecotype dominating over others. And this is why it's such a repeatable evolution. This happens across replicates. Okay, so that showed that there's evolution happening in real time in GreenNet, which is extremely cool. But is it driven by certain loci? Postdoc in our lab, Shingu, has created or utilized an approach based on this parallel signal across replicates to create a Manhattan-like plot where we have a p-value that is telling us how parallel the shift is across replicates in order to map the loci that might be adaptive in a given environment. And this, you can think of it as a extremely powerful bulk segregant analysis, right? Because we have 5,000 starting individuals in each of the replicates, and we pull information across replicates. So these p-values are very high, and they're Bonferroni corrected already. And we see some of the typical suspects, loci involved in flowering time and dormancy, but also very high peaks of loci that we're yet to know. So we're looking into that. But the power of GreenNet is not just that it is very high scale, that we have this unique evolving resequence over time data, but it is this. And I'll just show you pictures so that you can see it with your eyes. This is before and after snowing in that alpine region. So you probably guessed what the power is that we're conducting these evolving resequence experiments in extremely natural and sometimes extremely harsh environments that might reveal evolutionary trends that are not possible to recapitulate in greenhouse conditions, even if we had very sophisticated chambers. So we can ask the question, are populations evolving clustered by climates? So Shin utilized a diffusion approach of allele frequencies for each of the replicates that we had collected to understand evolutionary trends over time. And this is what you can see over 1 year, second year, and third year. You see that the different populations, right, are diverging over time, and they're colored here by clusters. And you can see that they're kind of going into different potential optima, who knows, which is extremely cool. So this is evolution, this is evolution happening in real time. But the next question is are they evolving to unknown optima? How are we going to understand these trajectories? Are those trajectories predictable? So we know that adaptation in previous experiments followed a local adaptation pattern. So we wondered, are maybe each of these populations readapting to the local environments, right? And are they using the standing variation of the genotypes that have experienced similar environments in the past? And how can we look at this? So as before, we can quantify whether each of these populations has been transplanted into a local environment, such as in this case, as I'm marking with my pointer, or faraway environment. So we can quantify the precipitation distance between all pairs of founder, that is wild population, to common gardens, which is hundreds of thousands of pairwise combinations of genetic distance. And then we can see whether those wild populations, those ecotypes, increase in frequency or decrease in frequency. And this is what we see. If the transplant distance is very high, either low or high, in this case it is in precipitation distance, to the common garden environment, the ecotypes remain at zero or just mildly increase. But the local ecotypes, you can see, that have been transplanted nearby, have shoot up in frequency, sometimes almost to fixation, suggesting that this is a very strong signal and predictable signal of local adaptation. And in fact, if you think of quantitative and population genetic theory, you can infer here a Gaussian distribution. So we're using stabilizing selection models to try to model the dynamics and the strength of these natural selection under quantitative genetic theoretical framework. So with that, we've seen that evolution appears to be predictable. Well, there's a lot of tests still that we need to do exactly how predictable, when is it predictable, when is not. We are continuously trying to map genes involved in climate adaptation. And some of them might be novel because nobody has done experiments in those conditions, for instance, high alpine conditions. And one thing that I think is a challenge for our community, population genetic communities, understanding whether we can predict whether a population will go to extinction using evolutionary models, evolutionary rescue models, which require blending evolutionary genetic processes with demographic and ecological processes and models. And I think that GreenNet will be a key resource for the community, which we're going to make available pretty soon, because in some locations, populations went extinct after a couple of generations, versus other locations, populations remain alive after half a decade. And this is a perfect scenario because we have demographic data and genetic data for both of those populations, those that went extinct and not. So this is a perfect toy data set for creating these new models and testing them. So from this section, we've seen that there's enough time for adaptation to take place, at least in these conditions with high standing genetic variation, although some populations went extinct and we don't understand what happens there, and we need to improve our modeling approaches in that realm. So in the last section, short section, I want to bring up the elephant in the room, which is if there's enough land, enough populations that we're maintaining across species to maintain high adaptive potential. So I want to bring to your attention my current worst nightmare. Hope you don't mind that I share. More than 50% of all species in biogeographic analysis seem to have lost some part of their geographic range, or their geographic range has shifted. And I guess that this would have a high impact on the genetic variation, right? These are kind of bottlenecks. And these bottlenecks have been quantified and are relatively substantial. The Living Planet Index that tracks about 5,000 mammal species, over 30,000 populations over time since the 70s, have seen a geometric mean decline of 70%. So what is the effect in genetic diversity? It's unclear. A number of people have posted that this is a high concern, and a couple of years ago, the United Nations proposed a preliminary target for the first time that was genetic in their global sustainable development goals, which was maintaining 90% of genetic diversity across all species, which is great, but surprising given that we do not know anything about the genetics of more than 99% of all species. So many people are taking the approach of sequence them all, right, over time to try to get at this. And we can do that generating GWAS panels like we've done in some model systems. And maybe we, genomic technology, the depreciation or the price goes down fast enough that we might be able to do this. But I'm not sure we will for millions of species, and we need to take decisions right now. So in our lab, we thought, can we combine biodiversity theory, where there's very sophisticated and some other pretty simple models on how species go extinct, and apply it to evolutionary genetics. So I want to bring you how some of the predictions of the number of species that might become extinct have been done as a background for how we think that we can make maybe predictions on how genetic diversity is being lost. The background of biodiversity theory is actually relatively simple. Much of the signal comes from a well-established law of the species-era relationship, which tells us that as we explore more area, or as ecosystems have larger areas, they harbor higher number of species. This species-era relationship follows a power law, where the number of species is proportional to the area scaled to a coefficient set that is 0.2 to 0.3. And this had been seen 100 years ago, and Preston officially derived it. And these are Galapagos Islands of different sizes. In the early 21st century, then, Thomas et al. Thought we could calibrate this species-era relationship across global ecosystems, and then, given that we know how much area we're destroying, maybe we can predict what is the next equilibrium of the total number of species in the future after that area loss. And this might seem like a very simple prediction. It's just a rearrangement of this equation with like a fraction of A. What will be the fraction of S? But it actually is highly used because it's very easy to apply, and it can be applied to very large global scales. So it's in IPBES assessment, so Intergovernmental Panel for Biodiversity and Ecosystem Service assessment. So I thought this might also apply to other levels of biodiversity, that is genetic diversity. So if we also know that when we explore an area, we encounter more and more genotypes, right? And some population genetic theory has dealt with this. And this curve might apply for each of the species in the ecosystem. But I didn't know any that was derived, any of such relationships that was derived based on area. So we used the Arabidopsis genomes to try to do, again, a thought experiment where, given the geographic distribution and genomic sampling that we have of the 1,000 genomes, so each of these boxes are population samples in Eurasia, we could draw boxes of different sizes and then just ask the question, what is the genetic diversity within each of these boxes? And genetic diversity we can quantify as segregating sites, which would be the equivalent to number of species, right? Allelic richness. And what we saw is that there is also a power law with the area of the boxes, right? So these are 1,000 randomly draw boxes in the distribution, all of these dots, and this fits a power law. By the magic of math, right? Log, then you get a straight line, easy to quantify slope, which is the coefficient. And very interestingly, this was a very similar coefficient, 0.3. So as you increase one order of magnitude, the area, you increase genetic diversity by threefold. And we call this the mutation-area relationship, and it's just a metric of population structure. So it's very related to isolation by distance, but is refactored into area and based on allelic richness. This can be derived mathematically, and we can talk about the details from population genetic theory. But I thought that I would bring a practical example on how this could be used. So this is an experiment, a simulation experiment, where we might ask then, well, if we have the area that has been lost for a species, can we predict the genetic diversity loss? And we can do this simulation where we might remove populations from the southern edge and move to the northern edge of the species, and then track genetic diversity in the 1,000 genomes, how much it has been lost. So by doing this simulation, contracting more the geographic region of Arabidopsis, what we see is that this theoretical line, which is the dotted line, and it's an extremely simple equation, right? Just the area loss and the coefficient that we had fitted, predicts very, very well the loss of genetic diversity. And just for comparison, this is not what we would have predicted from classic, we could say, population genetic theory. So with a bottleneck, we know that we contract the population size of a population, and depending on the starting population size, this is what a bottleneck situation would give you. But the mutation-area relationship captures the fact that genetic diversity is not distributed across individuals in the landscape homogeneously, right? We're not in a panmictic population, right? And if you remove certain populations like these isolated southern ones, you lose more genetic diversity than you would expect it under a panmictic population. This is very cool because then we could potentially apply this to many species. But we wondered, is this a general phenomenon or is it Arabidopsis specific? In a truth or force, we tried to ask this with as many genomic datasets that we could find. So our team started doing analysis for 20 species for which we found hundreds to thousands of genome sequence. And as you can see them in this map, highly dominated by Europe and North America. And we combined compiled up to or more than 10,000 geotagged individuals on these species. And this is in a cartoon representation, the results. So we fitted the mutation-area relationship for each of these species. And what we see is that the genetic diversity decreases with area loss, right? If we do the same kind of simulation scenario with differences that can be captured or absorbed by these scaling coefficient, which might probably tell us something about the strength of population structure, the migration rate, the gene flow that is species dependent and depends on some traits. But this is very cool because the coefficients were variable, but we can get an average of about 0.25 or 0.3, which could be applied across species if we don't have any genetic data. So we did this back of the envelope calculation, just very simple, just to get a rough estimate that give us a sense of magnitude of the impact of area losses. So if we take IPBES assessment reports or other types of reports, such as the Global Forest Watch, we know that we've already transformed 40% of the Earth land. If we plug that into the mutation-area relationship, it tells us that on average, an idealized average species must have lost about 15% genetic diversity worldwide, just from that shrinking of 40% of the geographic area. And this surpasses the preliminary targets of the United Nations Sustainable Development Goals. So in fact, I went to Montreal when these were described. There was a session about genetic diversity, and we were discussing that probably at this stage, we do not have the data to commit to a number, because the problem of putting a number that is unrealistic and is not data-driven and it's not solid is that it decreases trust of the population. It decreases trust on trying to attain those goals. So this wording was rephrased, which I was very happy about. But there's lots to do. We need to understand what are the countries that are most sensitive to these area laws and genetic diversity laws. Remember, we don't have genetic data for many tropical countries where there is the largest amount of species richness. We do not have very good comprehensive databases of per species geographic range shrinkage. And we also know that there's a difference between the immediate genetic diversity loss, which we have provided an answer for, and the long consequences that is due to genetic drift down the line, which we're currently working on. So with that, we've seen that many species are highly impacted because geographic distributions are being reshaped. And this means that we need to have aggressive policies to maintain enough land to maintain high adaptive potential. And evolutionary biologists and population geneticists haven't been typically in the table in these global biodiversity frameworks and discussions, but we need evolutionary policies. So even theoretical people like us that work also in model systems, we can make a meaningful contribution to helping developing these targets. This is because we know genetic diversity is key for rapid adaptation, and that likely many species have that, but we need to protect it. And with that, I want to thank all of our funders, the members in our laboratory, the colleagues in the Genetic Laws Collaboration, the GreenNet Network, all of you for your attention. And if you're interested in this science, please check us out and reach out. Thank you.